Welcome everyone to Wednesday Night at the Lab. I'm Tom Zinnan. I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension, Wisconsin 4-H. And on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight, it's my pleasure to introduce to you James Crall. He's a new professor in the Department of Entomology. He also has an appointment with the Nelson Institute for Environmental Studies. I'm about to ask him the 5 questions. James, where were you born?
Cleveland Heights, Ohio.
And where'd you go to high school?
University school, just outside of Cleveland.
And then where'd you go for your undergrad, and what did you study?
So I went to Swarthmore College, just outside of Philadelphia, for my undergrad, and actually my primary major was in sociology, which you'll see a little bit of connections here, but had a second major in biology.
Good.
Which became the rabbit hole I fell down.
And then where'd you go for your master's and PhD, and what did you study?
Yeah, I went straight into a PhD at Harvard University in the Department of Organismic and Evolutionary Biology. Organismic is a made-up word. The real word is organismal, but yeah, so basically big scale biology, not so much cellular biology, but yeah.
And then did you do any postdocs, or did you come directly here?
No, I stuck around. We had a kid right around when I was finishing my PhD, so we tried to stay in Boston. I was lucky enough to get to stay at Harvard for a postdoc.
Very great. And then when did you arrive here?
Just January last year.
Oh, good timing.
Yeah.
There you go.
Yep.
Ice fishing. Yeah. I arrived in Madison in January. It's not the best time of the year to arrive.
Yeah.
Well, I really appreciate this. We're looking forward to it. You're going to be talking to us about insects and ecosystem services on a changing planet, insights from the humble Bumblebee. Can we make sure his audio is okay?
They're working?
His microphone is good?
I think those are
very good. So I'm going to turn mine off. Please join me in welcoming James Crall to Wednesday Night at the Lab.
All right, thanks so much for joining on, not the most beautiful of mid-April days here, but I was in California for about 4 days. I heard I missed all the actual sunshine in the past month, but hope to see some more soon. Yeah, right. So yeah, thanks so much for coming tonight. As Tom said, my name is James Crall. I recently joined assistant professor in the Department of Entomology here. So I've been here just a little over a year, so it was really fun to connect more and tell folks a little bit about what I and now my sort of growing group are doing here at the university. And so in particular, I'm going to tell you about insects, how they're responding to some novel aspects of environmental change, and specifically what sort of broad insights we can take from one particular group of organisms that is near and dear to my heart, the bumblebees. And I just want to say I'm really thrilled to have been, I think one of the great things about UW-Madison is it's easy to recruit really great folks, so it's been really fun just quickly to sort of build up a group, and a lot of what I'm going to talk about today is I'm going to talk about it, and you're only going to see me here, but it's really a collaborative effort by my whole lab group. Okay, so I'm going to suspect that if you're here, you're engaged in science, you're excited about the pursuit of knowledge broadly, and I'm going to suspect that you probably don't need a lot of convincing that insects are not just sort of obnoxious little biting organisms, are actually critically important. So there are probably more than 5 million species of insects on the earth. I say probably because we've only actually described, so that means sort of taking the time to look at what makes a species unique and what the sort of particular combinations of body parts and morphologies are that are a species. We've only actually done that for about 1 million insect species, but we think that that's a tiny fraction of the species that exist on the planet. And those species are incredibly important for a huge array of really basic functions on our planet, right? So insects, of course, are herbivores. We see them in our gardens as pests a lot. We see locust plagues. We know that they transmit disease, so they do some stuff that we don't care particularly for, right? But also insects are the ones in some cases that are controlling those, right? So they're also predators that are controlling other insect populations. They're providing food directly to us sometimes, and increasingly so. And also, of course, down here on the bottom row, you'll see a bunch of pictures of animals that provide a service we particularly care about, which is pollination. So they pollinate flowers. It is important not just for biodiversity, but also for our food production. And so given that, there's real concern over sort of recent mounting evidence that we might be seeing declines sort of across the globe in insects. So this is just one picture from a study that came out a few years ago, which was one of the few studies to actually in the same place over a long time period with really careful methods monitor the abundance of all different kinds of insects. This is over the course of about 27 years in these nice nature reserves in Germany. And what they found actually when they went and surveyed those many decades of data is it looked like there was a really severe decline in the total biomass of insects. So maybe like a 3/4, about a 75% decline in the total biomass of insects. So pretty scary, and has really set off some alarm bells that even made it into some popular press. I don't know if anyone came across this New York Times Magazine article from a few years ago, but in the entomology world it's really made waves because we're actually making it to the cover of the New York Times Magazine. Not an everyday occurrence, right? But real worry about the insect apocalypse, which has sort of made it into our public consciousness a bit. And I think what we've learned now from a few years of really intense study of this is mostly about how little we actually know about this. So we may or may not be experiencing sort of catastrophic across the board apocalyptic conditions for insects, but there really is very clear evidence from what we have that across many different groups of insects, they are systematically declining in populations. But really what is highlighted is that we actually know remarkably little about the insects that are out there. So I told you a little bit about one group that is particularly important, that is not only near and dear to my heart, but that is really important for our well-being as humans, and that is the pollinators. So you'll see some pictures of bees in particular up on the top and bottom there. So we care a lot about bees and other pollinators because they're really important for just the functioning of biodiversity. So actually of all flowering plants on the planet, about 90-ish% are visited or depend to some extent on animal pollinators. So just the functioning of ecosystems is really dependent on pollinators. But also, of course, our food supply is also very dependent on pollinators. So roughly a third of global food supply comes from crops that depend on pollination from animal pollinators, or largely bees and other insects. And if we think about their sort of existing importance, we know that about a third supply depends on pollinators, but how much are we actually sort of seeing real world limitation of like how much is a lack of pollinators actually driving or limiting food production? And some really nice recent work has suggested that the answer to that is substantial. So if we look at basically how much yield potential we're missing, which you can look at by saying, oh, within a particular crop, what's the most you could produce on an acre versus the least you could produce in an acre? We call that the yield gap, essentially how much potential there is to improve yield. And we think about a quarter of that existing yield gap is actually due to lack of sufficient pollination. So this is sort of agronomically important. And we've also learned, so we mostly know about honeybees, right? They're the canonical, we all sort of know about it, and it's great to sort of have, especially backyard pet colonies, but also these industrial scale honeybee colonies that are really, really critically important for agricultural pollination. But there's about 20,000 species of bees across the planet, and that's just bees, not the other pollinators. And another thing we've learned is that honeybees can't do everything. And actually that diversity is really, really important for a whole bunch of reasons. And when I told you about a third of our food supply is dependent on pollinators, in some ways that almost could undersell their importance, because the crops that do depend on pollinators are largely fruits, vegetables, and nuts that are actually especially important not just for calories, but for human health. That's where a lot of our micronutrients come from. So this is just some work that came out a few years ago trying to estimate what would be the health impact of losing pollinators, and it's really substantial. Millions of additional deaths and sort of life years lost if we were to lose pollinator-dependent crops. So they're really important. What we want to know then is if they are declining, right, what are we doing in how we're changing the environment that could be impacting bees? Ideally with an eye toward mitigating some of those impacts, right? So we are changing the environment in lots of ways that are really critically important for bees. So one thing is, since the middle of the century, we've introduced lots of new pesticides and agrochemicals, including insecticides, but also things that aren't targeted at insects. Basically, the chemical environment, especially in agricultural systems, has changed a lot in the past 60 or 70 years. We've also, at the same time, of course, changed landscapes, right? So not only increasing the amount of farmland, or at least in the earlier part of the last century, less so now, but in addition to adding farmland, converting lands from, say, forest to farms, we're also, of course, and this is more sort of recently the case in our region, shifting how we use those landscapes from sort of small scale, maybe more diverse farms to more intensive, large scale agriculture. And those changes, and especially if we think about the sort of flowering resources in those landscapes, those look very different to a bee, right? So what the flower species that are in those landscapes can really impact what's happening to them. We're also introducing novel disease, just in general by moving, increased global movement, but also specifically by transporting honeybees that actually vector a lot of diseases that can have impacts on wild bees. And of course, we're fundamentally changing the abiotic conditions of life on our planet. So all of these sort of novel environmental changes can impact bees. They can act alone, but also critically we know and increasingly know that these can act in concert. So sometimes it's not just the sort of one new stressor. It's not just the pesticide. The pesticide might interact with the food available in the landscape and these sort of death by a thousand cuts kinds of impacts that can be really challenging to study. So what we want to understand is, how do these ways in which we have impacted the environment, what do they do to, again, bees? And I'm going to focus a lot on my, again, particular favorite group of pollinators, the bumblebee. And so one thing we want to do is think about what that does to sort of single organisms, right? Think about that individual bee that we see outside. And one thing I want to do today a little bit is convince you of how cool the bumblebees are. So I chose, I mentioned humblebee in the title of this talk, and partially that's because I think bumblebees can get a little bit of a bad name. And actually, during Darwin's time, there were no bumblebees. They were all called humblebees. And this is a great quote I found on an article describing that change. She described it as the clumsy, when they started becoming named bumblebees, the clumsy-looking furry bee with its pitifully small wings and tubby body was the perfect match for its new, slightly belittling name, as it bumbled from droopy bloom to droopy bloom. And so sometimes a lot of my early life, and the reason I sort of came to bumblebees was actually I was trying to study some aspects of how they fly, which is really fun. So I spent a long time flying them in wind tunnels. And they certainly can look clumsy sometimes. They bang into stuff all the time. Head first, sometimes they bang into the same thing over and over. So, in some ways I don't blame folks for having that opinion. But they're also totally remarkable organisms. So just to give you a quick sense of this is a sort of typical day in the life of a bumblebee and where we would often see them. This is on a windy hillside in Vermont in this case. I'm just going to show this again, highlighting something you might have missed. And that is a single bumblebee forager out, moving through this environment, trying to grab food resources from these flowers, right? So this is, in some sense, not that surprising. This is something we see pretty regularly. But I want you to try to think on the scale of a bee for a second and just think about how amazing this is. If you imagine like a human helicopter trying to maneuver through skyscrapers that are flopping back and forth really quickly, right? It's an incredibly dynamic environment, both in space and time. They're doing that with a few orders of magnitude fewer neurons than we have in our brain, and they're doing it with all the energy they need to do that for hours and hours, strapped onto their tiny back on about 250 milligrams. So, they're amazing physical, physiological, and sort of biomechanical and behavioral organisms. The adaptations they need to move through this environment are amazing. But with all that adaptation, to the external environment is actually aimed at is something that we usually don't see. So I want you to just train your eyes on that little hole. So what you're going to see is a bumblebee forager come in, loaded with pollen, and then go. This is like a very hard act to actually see in the environment. But so what those bees are doing when they're out, having all that physiological adaptation to flight, what that's for is bringing resources down to the nest, which actually in the case of bumblebees is underground. So this is a look at what happens, quick little glimpse into what happens inside of a bumblebee colony. So bumblebees are social, right? So they don't have the tens of thousands of workers that a honeybee colony has, but they have usually up to maybe 100, couple hundred workers, but they're eusocial, so most of them don't reproduce. And when that food comes in, other workers are sort of taking that pollen and nectar being brought in by foragers, and then actually reallocating that and sort of feeding that into where you're seeing these pods underneath are developing larvae that these other workers are cleaning, opening up to put little bits of food into, actually incubating. So they basically warm their young, and we'll do it collectively. I'll show you some pictures of this later on. So there's this whole sort of hidden world of growth that is the social life of bumblebees. So part of the reason I show you that is we often think about the aspect of that cycle that we see outside a lot, but what we really care about in some senses is not what happens to the individual bee, but how those impacts on the individuals scale up to the sort of function and ecology and reproduction of the whole colony. Because in some sense, the individuals are just cells in the body, right? They really are a superorganism in the sense that the whole colony is what reproduces. So if we want to understand the health of the whole colony and the populations, we need to understand what happens at the colony scale. On the flip side, we want to not just sort of go from the individual to the colony. One thing we're realizing more and more is it's sometimes not that simple. Sometimes what the colony is doing can actually feed back into the individual. And so what I mean by that is, you might sense that sometimes your behavior is a little different when you're around other folks, right? You're responding to all the social stimuli in your life, right? And sometimes maybe that'll affect, if you have enough adrenaline from an exciting conversation, maybe that adds up with the coffee you had, right? So that the external stressor might actually be like affected by your social environment. And we see some of the same things happening in bees. So we, again, want to understand not just what individual impacts, how those scale up to colonies, but how what the colony is doing sort of feeds back to the individual. So what I'm going to focus on in particular today with some connections to these other aspects, but I want to dive in a little bit and tell you about some work that I and my group have done trying to specifically understand the impacts of novel pesticides and agrochemicals on these sort of individual and collective behaviors in bees. And in particular, I'm going to focus on neonicotinoid pesticides. Has anybody heard of, raise your hand if you've heard of neonicotinoid pesticides? Okay, so good. So at least that word, and it is a mouthful for sure. Well, I'll start saying neonics after this probably. But these have gotten a lot of attention over the last decade or so. These are a very widespread class of relatively novel, although not so novel anymore, insecticides that work by targeting the nicotinic acetylcholine receptors. What you really need to know about that is this is the primary excitatory neurotransmission system in the insect central nervous system, so it's a bit of a brain sledgehammer in some senses. And since their introduction in the mid-90s, which is probably too small for you to see, but that's over 1994 is the left of that graph, the use of these compounds, and there's a few different specific compounds, has just taken off across the globe. They're now the most common group of insecticides used globally. And since their introduction and now with sort of increased monitoring efforts, one thing we've realized is they are remarkably persistent and remarkably widespread in the environment. So one of the things that a particularly widely used or avenue of use for these compounds is to actually treat seeds of crops with them. And so what they do is they get taken up systemically by the whole plant when a seed is treated. And so if you're a grower, that can be really great because that compound shows up in all the tissues of that crop sort of throughout the growth cycle. If you're a bee, one of the downsides of that is those compounds show up throughout the plant, and that includes nectar and pollen resources that a bee might be feeding on months after the initial application at the seed. We also know now that they accumulate in soil over years. They can show up in groundwater, a sort of enormous array of environmental impacts of these compounds. Just to give you a sense of actually how widespread the exposure to bees is, this is a great study from a few years ago, where what they did is sort of like the most delicious sounding experiment of all time. What they did is ordered as much honey as they could from as many places across the globe. And so every dot there is a honey sample. If you think about, honey's great because it's food for us, right? But it's also kind of an environmental sample in some ways, right? It's the bees going out and in their environment, going and foraging from whatever floral resources are available, right? So this is kind of a cool way of getting a look into what are the bees actually feeding on and what does that tell us about the nectar contained in flowering plants across the globe. And the answer to that is that there's a lot of neonicotinoid pesticide across the globe. So the white samples, white dots there show samples that did not have detectable neonicotinoid. That's the minority. It's about 25%. So the majority have some degree of neonicotinoid present. Actually, many of those have multiple compounds and often at exposures that we know have impacts on bees. And now there's really very strong and clearly mounting evidence that at field realistic concentrations, these compounds can impact bee colony growth and also abundance of wild bees. And we also now know that they can impact other they can sort of have these cascading effects. So for example, one thing that they can do sometimes is crash invertebrate populations in even freshwater systems. This would be obviously of concern around here. That can impact fish and other vertebrate populations. So a lot of where my work has focused is trying to understand really what the mechanisms of those impacts are, with the idea, and I'll tell you a little bit more about this later on, with the idea that if we can better understand how those impacts are happening, it might be able to help us better mitigate those impacts and also potentially design alternative routes, and better management practices for those compounds. A lot of what we do know is about, again, that aspect of the bee life cycle that we know well, foraging outside. So there are important impacts on being able to navigate to flowers, learn floral resources, and sort of bring food back to the nest. But where a lot of my work is focused on, well, what happens to that social life that I was telling you about? Once those compounds once the nectar and pollen are brought back to the hive, those contain pesticides, right? So what happens to all the sort of social life within the nest? And so just a quick outline of some of the results or some of the work that I'm going to tell you about. I'm going to give you a quick sort of introduction, or a little more of an intro to the social life of bumblebee colonies, and then tell you a little bit about how neonicotinoids impact that. Some more recent work trying to understand both combined effects of neonicotinoids and temperature, and then tell you a little bit about more recent work. Okay, so bumblebee life cycle. Has anybody seen a bumblebee yet this year? Probably not. So we're coming up on probably a few weeks away. So one of the really interesting things about bumblebees compared to honeybees is they're social, but they actually start their life off kind of like most insects, alone in a hole in the ground. So they actually start off solitary. So in the coming weeks, you'll start seeing bumblebee queens emerge. So they come out in spring, have to do their own foraging, and will start their own nest. They'll lay eggs that will then eventually develop into workers, and those workers sort of by midsummer, so around here this will be sort of mid-May, late May, that workers will have come out, so you actually won't see the queens anymore, not because they're gone, but because they're underground, basically focused on egg laying. So only in the next sort of month and a half or so will you likely actually see most species of bumblebee queens coming out. During summer, they'll sort of grow, most of the bees in the population will be sterile workers, and by the end of the season, the whole colony will get big enough that it'll send off, the sort of colony will reproduce and send off workers and males that will then mate. All the males die, all the workers die. New queens will save sperm actually from usually that one mating, go back into the ground, wait out winter until the time of year comes to start a colony again. Any questions on that? I should say I'm also happy to, feel free to interrupt me if I say things that are particularly nonsensical or particularly interesting. Yeah.
But so how long does the individual bee last? Is that all summer?
No, it sort of depends a little bit on what you're doing. Foraging is really dangerous. It's really energetically costly, and also, there's predators out there, all kinds of stuff. But so there's actually kind of not really great estimates. They live a long time in the lab. In the wild, we don't really know quite so well, but probably like 5-6 weeks, something like that. And the whole colony is a couple months.
Are you going to tell us how to attack?
Yes. So this is a view inside of a bumblebee colony. So this is what it sort of looks like. Not the beautiful sacred geometry of a perfectly aligned honeybee colony. They're kind of like the slightly sloppy cousins. They are actually the evolutionary cousins of honeybees. Yeah, and so you notice that they have some very strange markings on their back, which are not natural variation. So what those are some, basically a system I originally developed during my PhD that let us develop really simple little barcodes. So it's kind of like a QR code, but with a lot less information on it and made very small so that you can look in a photo or a video and actually identify what individual bees are doing. So what's fun about that is you can then tag all the bees. So now that's looking at that same scene within the bumblebee colony. And now with video, we can sort of track automatically. So Tom was talking about, you're going to hear about networks next week. So this is I'll talk a little bit about bee social networks a little bit today. But so what we can do is track the sort of movements, activity, and also social interactions of bees across these whole colonies.
How difficult is it to physically put those tags on?
It takes a little bit to learn, but it's pretty easy. So we have ways of sort of anesthetizing them in the lab, and then you'll be shocked at the detail, which I noticed. When I first started off, it took me about 3 minutes a bee. It takes me about 25 seconds a bee now. Just really, the mechanics of it you get used to, right? It's like any little lab skill. But yeah, so we can do this, and what's great now is what used to take us just an enormous amount of labor by monitoring by eye, now we can do pretty automatically, which is really fun. So what do we learn from this? One of my first things, which is actually sort of where my research started in this area, is looking at individual variation, right? So you look at a bunch of honeybees or bumblebees, and you wouldn't think like, oh, they have personalities, right? Or they have variation. But it turns out there's just a remarkable amount of individual, repeatable variation that particular workers do particular things sort of over the course of their lives. And a lot of it we don't really understand. So one way we know that is by doing that automated behavioral tracking. We can do things like just sort of track what behaviors bees are doing. So this is 4 sort of behavioral categories. You can be foraging, you can just be hanging out on the side of the nest, you can be sort of moving around and patrolling the outside, or you can be actively nursing. And this is 2 days in the life of one single bee. And so what we see is this is a bee that liked to forage, but if we do the same thing across multiple bees, what I want you to see is toward the top there, that was a bee that really liked to forage and did that from day 1 to day 2. The second from the bottom, we had a bee that liked to do nursing and consistently was doing nursing from day to day, and on the bottom, a sort of active patroller, or cleaning bee. So this, so in many ways what we mean when we say in humans, right? So they're taking care of the developing young, feeding them, incubating them, are the sort of primary, and cleaning and some other, but those are really the big ones. So they are very actively, they're not like other insects, or at least some other insects that sort of leave eggs and a load of food and then say bye-bye. They're actively taking care of so one thing they do is called progressive provisioning. One of these sort of hallmarks of sociality, they're continuously feeding their young. So there are workers that are largely devoted to staying inside of the nest and yeah. So they sort of vary individually in what particular behaviors they like to do, but also they, interestingly, vary in where they even like to be within the nest, even for the ones that just hang out in the nest. So how do you know that? So you can imagine we can do that tracking for, say, all day long, those little, scurrying bees that just looks like total chaos, and it doesn't look like, kind of hard to pick out patterns from that sometimes, right? But if you watch that for long enough, say over the course of a day or multiple days, you can get a sense of, okay, where did this particular bee like to be? No pun intended. So you can sort of see like there's a home range for a single bee on a single day. And this is that same bee over 2 other days. So you can see, this is sort of where the nest isn't shown here, but this is like where the nest stuff is. So, that bee is probably nursing a lot, spending a lot of time right on the nest structure. Well, maybe that's interesting, but maybe that's just where all the bees tend to be, right? But if we do the same thing for a few different individual workers, what you see are pretty strikingly individually unique patterns. So a bee that really like to kind of be on one particular part of the brood, one worker that really like to be on the outside of the nest, and that occurs repeatedly across days. So they have sections of the nest that they like to hang out in. And I won't go too deep into this. This is one of the more in the weeds slides I'll show, but it's kind of fun. So one thing that we can do with this system is we're monitoring workers sort of continuously from day to day. We can do that passively, but we can also mess with them. Right? So one of the ways we can mess with them is by looking at, this is darker colors here are showing you bees within a colony that tended to be more active foragers. And they, again, do that repeatedly day to day. We can wipe some of those active foragers out and then see what the colony does. And essentially what they do is respond by both having some sort of low activity workers increase in how much they're foraging, and then other bees that weren't foraging at all can start foraging. So it turns out, and this happens within about a day, so they can sort of actively reallocate their task force. They're very efficient, much more efficient than we are in some ways at reallocating workers when foraging work needs to be done. One of the fun things about this is we can look before and after, and there's a bunch of bees that I'm not showing you down here that never switched. I'm kind of showing you the switchers here. So we can also ask, what made some bees switch and not other ones? And basically the short answer is that bees that tend to be more toward the middle are more likely to make that switch, and we think that has to do with information flow. So if you think about where, if you're trying to figure out, make a decision like, oh, should I go to the grocery store today, right? Should I go forage? Where are you going to get that information about whether you need to go to the grocery store, right? It's at a particular place in your house. It's probably in the pantry, in the fridge, right? Well, the pantry and the fridge for bees are in the center of the nest where all their food reserves are, and maybe also where other bees are that might be able to communicate some of that information. So maybe where bees are in the nest determines what kind of information they get, and then actually determines how they make some of these decisions about sort of task switching. So that's just a sort of quick intro to social behavior in bumblebees. Now what I want to turn toward a little bit is thinking about the impacts of neonicotinoid pesticides. How do pesticides disrupt that system? So one trick we can pull is that we can actively feed. What's nice about bumblebees is you can pull them out, actively feed them sort of controlled amounts of nectar, and then put them back in the hive and see what they do. So what we can do is actually use this and combine it with the tracking I just showed you to study the impacts of pesticide exposure, where we can, within a single colony, pull out workers and either give them a control, so just regular sucrose solution, a low amount of imidacloprid, which is a really common neonicotinoid pesticide, or a high dose. And both of these are sort of within the naturalistic range. Basically, these are the amounts that we think bees consume in either a single foraging bout or a single day of foraging. So what we really want to know is, now, given what I've told you about that social system and all those important social behaviors, how does pesticide exposure impact those behaviors? So we can now look before and after that exposure and look at, for example, the change in how much time bees spent nursing. So before and after exposure, what we see is that for control bees, so bees that just got sucrose, luckily we don't see any effect. So, it's kind of, we pull them out of the nest, we mess with them, we starve them, we put them back. Luckily, that doesn't seem to have an effect. So just the sort of handling doesn't really impact them, nor does our low dose of imidacloprid. But a high dose of imidacloprid significantly reduces the amount of nursing behavior within the colony. So we see a reduction in the amount of time that bees are spending nursing. We actually see similar effects on activity, so they're moving around less within the colony. They're further out from the social center of the nest, which, I just told you is actually pretty important. Where they are in the nest might be really important. So we see these sort of spatial dynamics impacted. And now I get to my one social network graph as a preview for next week. So what we can do is we have a map of when bees physically made contact with each other, right? And that is actually a really important form of communication. And so we can use that to make our own little sort of Facebook of bees. And the way we're visualizing this is individual dots here are bees, and two workers are connected by a line if they physically interacted during the time when we were watching them, right? And then the red dots are workers that were fed our higher dose of imidacloprid. And the graph here, what it's trying to do is basically push less connected bees to the outside. So the main thing to see here is just that the red dots really tend to be on the outside. So they are on the periphery of the social network. And we see that reflected in just a measure of overall number of interactions. So they have less social interactions. So the takeaways from this are less nursing, lower activity, further away from the nest center, and less interactions with their nestmates. So we think, see really important effects of this common pesticide on social behavior within nests. So those experiments were really the sort of first step we took in trying to understand what these compounds do to nest behavior. But one of the challenges is it's still kind of unrealistic, right? You can still look at that and say, well, being pulled out of your nest and being force-fed a syringe of, this amount, it's, maybe it tells us something, but it's not really that naturalistic, and it's not how bees would normally be exposed to this, right? Which is sort of continuous low levels of this. So to study those impacts, one of the challenges is you can't really do that sort of individual scale exposure where you can use individuals within a colony. And for some sort of nerdy statistical reasons, it makes it very challenging to actually use individual workers or individual bees as your sort of statistical replication. So then one of the big challenges is, well, then you have to replicate it whole colonies, and that is a pain in the you-know-what, right? So taking the kind of tools I just showed you, but scaling up to whole colonies as your individual replicates is really challenging. So this is a lot of what I actually focused on for my postdoc, is trying to take those tools and scale them, use really kind of innovations in robotics to take those tools and take them across colonies. So this is actually a benchtop imaging system where we can put multiple individual little bumblebee colonies on, and then within each one do the same kind of tracking I just showed you. This is what it looks like in the lab. It always looks a little sloppier in real life. I'm hiding most of the duct tape, but not all the duct tape that was in there. And this is what it sort of looks like in real life. So each one of these little boxes is a bumblebee colony, and then this is a robotic gantry bringing cameras into place over top of a colony and then moving on to the next colony. And so this is just a time lapse of that system running continuously sort of 24 hours a day. And what's nice about this is now we can start getting up to the level of replication where we can use sort of whole colonies. So we can collect trials about 12 times a day for 12 colonies in parallel, and now we can do this sort of more realistic whole colony exposure to pesticides. So what did we learn? These are videos from that system of bees immediately after exposure. On the left, these are control bees, and on the right are imidacloprid exposed bees. So not any super striking differences to the eye. Colonies always look a little different, so right after exposure, we don't actually see particularly strong impacts. But these are the same colonies about 3 days later. That is actually a video. You can see one bee running around. But one thing you might be thinking to yourself, like, do you really need a fancy robot to tell you that that those look different, right? It's a pretty striking, like, you know, nobody's moving, everybody's moving, right? But this effect has actually been hidden from us in a very interesting way, and I'll explain that in just a second. So if we use this system, like I said, we can record continuously sort of throughout the day or over multiple days. So if we sort of watch what these colonies are doing for about a week, and then just quantify the sort of average amount of time that bees are spending moving. So this is the overall activity. What we see is kind of what you'd expect to see. Pretty high activity, you know, these colonies are always moving, but with a little bit of a dip overnight because there's a little bit less foraging, and a little more nursing, and sometimes bees sort of relax a little bit overnight, but pretty high activity. And these are our 9 control colonies. If we compare that to our imidacloprid exposed colonies, what we see is an overall reduction in activity. But what I want you to see here is that actually the effects are particularly strong overnight, which is those gray bars. So the colony I was showing you that looked just totally like comatose is actually in the middle of the night, and that colony during the day looked basically totally normal. So they sort of rebound during the day. So for some reason, these impacts are showing up the most strongly overnight and not during the middle of the day. We see this consistently across colonies. So now if we look back at some of those same measurements we took before, we see that actually during the day, we don't see any effect on nursing between our control and imidacloprid exposed colonies. We do see a significant reduction in nursing overnight. So we see a similar pattern for those spatial dynamics. So again, we saw them move toward the periphery of the nest. We see that show up overnight, but not during the day. And overall social network connectivity, so how much they're talking to each other, reduces overnight, but not during the day. So I think just the takeaway from this is we do see we see a lot of the same effects showing up, but they have this interesting and sort of strange rhythm where they get strongest overnight and then come back during the day. And so that's all in the lab, right? Which I think tells us a lot. So we've gone from, okay, the most artificial sort of feeding a bolus of pesticide to individual bees to slightly more realistic sort of look at whole colonies, but that's still in the lab, right? One thing we wanted to think about, well, what does this look like if we move out into the field? Can we study some important aspects of social behavior out in the field? But man, the robot does not like to go outside, right? So for reasons you can imagine. So one of the nice things is there's a really important social behavior that we can study outside without having to use a camera to watch colonies. And sorry, this is not showing up enormously well, but on the right there is a thermal image of basically the same scene on the left. This is inside of a colony again. And they are, I told you before, you know, when I say nursing, one of the things I mean is they're actively regulating temperature of the brood, and they actually do that collectively. So they get together, heat their thorax up, which is all their flight muscles, shove that hot insect blood, hemolymph, into their abdomen, and then hold that abdomen against the developing young to incubate them. And they do that all together, sort of as a ball of heat-generating bees. And so what we can do is put colonies outside and then basically put temperature probes in the middle of the colony and see how well they're doing that important behavior. And so we can track the temperature. This is outdoor temperature for, again, over the course of about a week. And then green there is a control colony. You can see they're doing a pretty good job. You know, there's some fluctuations, but they're doing a pretty good job keeping that temperature within a fairly narrow optimal range. But our imidacloprid exposed colonies, we see, you know, they're still keeping it above ambient. They're doing, you know, they're doing some thermoregulation, but they're not doing as good of a job keeping it in that narrow optimal band for sort of homeostasis and colony growth. And so it does, in fact, in the field sort of impair social thermoregulation. One of the last effects I'm going to tell you about that I'm happy to talk about more, but I want to get to leave a little bit of time at the end for thinking about things we've been telling you a little bit what we're doing here. But it's another remarkable thing bumblebees can do is actually adaptively change their nest architecture. So this is what they look like sort of when we order them from the internet. You'll see sometimes they start to build up kind of a wax canopy or a little cover over the top of the colony. And sometimes this is just like a whole cover, and basically it works like a blanket. So what they're doing is creating, they're getting rid of the drafts in the house, right? They're closing it up and keeping that sort of thermoregulatory layer or building that on top of the colony. But what we see is colonies that we put outside, those same colonies that we were sort of monitoring temperature in, seem to totally lose that ability to adaptively restructure the nest. So, and this is a behavior that sort of occurs over a week or two often. It's a very slow behavior. So at least impairs, possibly totally eliminates the ability to build these adaptive nest structures. Okay, so these last two bits are going to be much quicker than the first two. Any questions on there? I'm happy to stop. Cool, well, follow up. So what I want to do a little bit now is think about the other direction of that arrow. So that told you a lot about, okay, what's happening to individual bees, what does that do to the colony? But now I want to real quickly think about what is the colony state, how can that maybe modulate some of the impacts on individual bees? And so if we come back to this life cycle of bumblebees, one thing that's kind of unique in this system and is really interesting is there's actually a solitary phase and a social phase, right? So during the course of summer, they're growing from just a few workers, or really one queen initially, to a few workers, to hundreds of workers. So there's this sort of gradient in sociality during the summer case here. And there are some good theoretical and empirical reasons to think that sociality might actually be an important buffer for, right? Because, we think that these big complex systems might be more robust in some ways. So for example, from some really nice field studies that have looked at the impacts of pesticides, one thing we find is that there's very strong impacts on wild solitary bees of pesticide exposure. There's often sort of moderate impacts on bumblebees, and maybe sometimes immeasurable effects on honeybees. And again, these are colony size of 1, colony size of a couple hundred, and colony size of tens of thousands on the right side. So this suggests that maybe there's some aspect of being in a social system that buffers the impacts of these compounds. And so to get at this question, what's great about bumblebees is we have that variation within a species, so we can actually use it as a sort of experimental system. By taking colonies, bringing in queens from the lab, letting them develop, and then at different times during colony development, randomly exposing them to stressors. And either a control exposure, just pesticide, a cold snap, or a cold snap and pesticide. And this is what our cold snap in the colony looks like. This is again a thermal imaging camera. You can see that sort of hot ball of bees when things are getting really cold. That's them clustering around the developing brood. And so if you measure, for example, the distance between bees, so how close are bees getting together, you can see that when things get cold, they do sort of cluster together, and that actually gives us a measure of how well they're doing this sort of adaptive behavior. And one thing we find experimentally is that indeed these young, small colonies are actually much more sensitive to the combination of pesticide and cold than larger colonies. So, oh yeah, and this is, just one last thing, and this is actually things we've been doing more recently here. Young colonies are also different than larger colonies for a whole bunch of reasons. So that experiment was done on sort of naturally developing colonies. But one thing that we can do, which is fun, and I'll tell you just a little bit more about this system, but I want to get the picture in your brain, is that we can also sort of make our own little boxes that the electronics and ability to do this is getting really cheap and scalable, which is fun. And so what we've recently done are some experiments where we manipulate colony size instead of letting it change over time. and we show that we can basically recreate the same result. So it does really look like just smaller groups are more sensitive than larger groups. Okay, so in conclusion, overall we find that there's this interesting variation between individuals. We do find that neonicotinoid exposure impairs these really important social behaviors within bumblebee colonies, but that those, again, can have this sort of odd circadian variation where overnight we see much stronger effects than during the day. We also find that in addition to their direct effects, there are probably interactive effects. Remember I said, oh, all those multiple aspects of environmental change combine and they don't just work alone. So what we see are at the very least interactions between temperature and neonicotinoids. And also that colony state can maybe modulate some of these impacts, and in particular colony size. So there's some really fun and really interesting like basic biology questions. But one of the points I want to emphasize here is if we think about the practical implications of some of these compounds, generally when we decide whether or not a pesticide is safe, even if we're doing lab studies, which was a big push to even get impacts on bees sort of on the docket, right? We almost always do those tests, and this is true for almost all experimental like behavior. We do it in the middle of the day in a nice warm room, usually in well-fed colonies, that in the case of bumblebees are often like at late colony stage, right? So our results suggest that that is like the worst possible combination of things to actually understand what these compounds are doing in the very challenging, very real conditions outside of the lab. Okay, how are we doing on time? Maybe I'm going to spend just like 2 minutes, and then I know we want to leave some time to just chat. Telling you a little bit about some sort of ongoing and near future work. So in particular, Tom asked me to talk a little bit about a grant that we just got funded that we're really excited about, this very generous funding from the USDA. In brief, trying to build, so one thing you'll notice is a lot of theme of what we do is basically using computer vision. We take a lot of pictures and videos of bees and try to extract information from that. And I think it's a really exciting time to be doing that because the sort of technology for doing that, the camera technology is getting really high resolution and really cheap. It's in our phones, it's in every device around us. At the same time, the tools for taking information out of those videos is also moving really fast. So in general, what we're really interested in doing is, and what we've gotten some, again, very generous funding from the USDA, to work on is try to adapt some of these tools for better understanding, in particular, bumblebee health. And this work that is sort of, we're in the very early stages of, just kicked in a couple months ago, but it's going to work sort of across scales, and one sort of try to, in the lab, bring some more of that environmental complexity into the lab a little bit, and develop systems that can help us better screen not just single compounds, but lots of these different compounds sort of in parallel, and use the fact that we can do that on really cheap electronics. And then go from that sort of high throughput screening in the lab out to the outcomes that we care about in the world. And one of those outcomes that we really care about, obviously, is the actual delivery of pollination services. So what bees are doing on flowers, and also, of course, colony growth. So taking these tools, I said, the robots generally aren't happy very outside. We're trying to take the robot, not actually the robots, but we're taking some the computers outside to try to understand what some of these more real world impacts are. So for example, I already showed you these pictures, but these are some prototypes where we can use sort of 3D printers and laser cutters and very cheap electronics in the lab. These are Raspberry Pis that normally cost about $35 right now because of supply chain stuff. It costs about $200. But luckily I've got a stash of them in the lab. But so we can sort of build these very cheap units that instead of costing the few $1,000s it did, like when I was doing my PhD, sorry about that, now it costs a couple $100s to build one of these units, so we can sort of use these systems to really do things in parallel. And then also, I don't know if you're going to be able to see this, but we can also do really fun things like, in addition to just where tags are, we can now track individual body parts using some sort of deep learning and new really sophisticated computer vision tools. So what we want to do is use those to better understand, and especially sort of at scale, what these impacts are. But we're also really interested in, again, moving out to the outcomes that we care about, like the delivery of pollination services. So in addition to what we can do sort of in hives, there's some really exciting tools, and we're helping sort of adapt them to the system where we can automatically monitor visitation at flowers by bees, and even also do things here like track their body parts, maybe even start getting into questions about pollinator efficacy, why different species sort of do different things, do they interact with flowers differently? So some fun questions there, and again, really taking advantage of this low-cost scalability. And this is actually out at Arlington Ag Station. In particular, we're working on trying to see not just impacts within colonies, how does pesticide exposure impact the delivery of pollination services and sort of experimental systems. There's a couple of folks in the lab working on this. And then finally, this is sort of the most fun in some ways because it's where we all start as biologists, is thinking about what these organisms do out in their environment. Bumblebees live in some really wild, challenging environments. So this is actually a very typical day in the life of bumblebee. In addition to what I showed you before, this is a bumblebee queen trying to start a nest out on top of the Continental Divide at about 12,000 feet. This is like par for the course for them. They have just a remarkable sort of global distribution. So we want to understand what some of those real world stressors look like and what we can sort of learn from the lab, but take that out to what colonies are doing in the field. And so in particular, we're working on, again, taking these same tools, putting them out in the field, trying to understand the impacts of real landscapes on colony behavior and growth. And again, one of the most fun things about this is we're working on everything I told you about today is one species, Bombus impatiens. This is our sort of common species around here, but there's about 250 species globally. Some are alpine specialists. Some we actually just got back from a trip visiting a group working on the Mexican bumblebee. There's single species that run that whole gamut. And we can bring a lot of those into the lab to study them. So really fun to think about moving back out into the sort of evolutionary and ecology context. All right, and with that, I just want to again say what I told you about is the work of many, many folks and has been generously funded by many groups. So thanks again. Sorry that might have run a little long, but yeah.
Great. Once again, if you put the microphone right next to the phone, it picks up the chat. Sorry about that, everybody.
That's all right.
Key facial recognition of flowers versus your computer
Here you go, Sandy.
camera recognition. Please.
And so a bee's ability to recognize.
Well, yeah, okay, I'll repeat the question. I wonder if you looked at the bee's perspective of facial recognition. If they get the nicotine, that they no longer can recognize a flower, and how that affects their ability.
Yeah, that's a really, so there's a couple aspects. I don't know if this is where you're going, but like social insects are wild. We don't know this so well for bumblebees, but like there's a group of wasps that have variation in facial markings. They actually know each other by face, so they learn each other's faces. We don't really, there's not direct evidence for that in bumblebees, but so one thing that might be sort of impaired is their ability to actually sort of, recognize each other or mediate these social interactions. That could be a part of what we're seeing both in the nest, but also certainly we know that there are regions of the brain, particularly with having to do with sort of learning and sensory integration, that are impacting the ability of bees, not necessarily to learn sort of individual flowers, but definitely impairing their ability to learn what flowers have good food. And so actually one of the things I skimmed over very quickly, but one of the things we're very interested in with our flower visitation experiments is not only looking at the impacts of pesticides under sort of normal conditions, but let's say you combine that with like a drought or some shift, right? Where normally we think the bees are really good at sort of adaptively learning that and figuring out, like, as food resources are shifting, they're remarkably good at figuring out what has the good food on what day. They're like excellent foragers, right? But under those sort of challenging conditions where things are changing really fast, do neonicotinoids impact the ability to do that rapid learning? So I think that's a really great question. There's definitely good reason to think that is part of what's happening.
And then a question from online. Do queens live longer than a year? What's the life cycle and how old is a bumblebee?
Yeah, so they don't do the thing, with possible exception of some Mexican bumblebees that have a bit of a different life cycle that's more continuous. Like, they don't do what honeybees do, where the same queen lives year to year. So basically, the queens that are emerging now, or in the next few weeks around here, will start a colony, be around for the whole growth of that colony, but then new queens will emerge in the fall, mate, overwinter, and start. So the original queen does not keep that colony going. They're basically annual. You can think of them as like the annual plants of the social bee world.
So
Yeah.
the queen dies in the fall, the original queen? Wow.
Yeah, so about a year. I mean, give or take, but yeah.
Aren't there some cases where the queen is killed?
Yeah, I was just wondering, on the subject of queens, I thought I read somewhere a long time ago, there's instances where they kill the older queens.
Yes, so there's a whole bunch of interesting, they can like revolt against the, the queen and basically take over. And we think there might be a bunch of reasons. Essentially, the workers have their own potential interest in reproduction, right? So normally they're sterile, but that's actually a little bit of a conflict, right? So most of the time that system runs well, but not always. And so, yeah, there's definitely some really interesting.
There.
Is there any research on whether the neonics
Yeah, good. That's where I get
applied
to. Yeah.
to a crop the bees don't actually pollinate, like wheat, get into the weeds of that field that the bees do visit? Do we know if that particular route of neonics to the bees actually happens?
There is now very convincing evidence that happens and is a very important route of exposure, yes. So if you have flowers growing in a field margin and you exactly plant wheat, or exactly, that gets into the groundwater, probably via groundwater movements, possibly some other routes, but yeah, they will definitely show up as residues in adjacent, not directly treated plants. So, yeah.
Online, do bees impacted by neonics hatch fewer eggs, and does the pesticide get passed along to the young?
Oh, that's a great question. Yeah, so the young within the colony, it definitely does. So there's a whole bunch of, what I told you is in some sense just a subset of what we know of the whole impact. So we know it impacts egg laying. There's really well-established impacts on sort of long-term growth of the colonies. Part of that might be things like, all of this stuff has to work well for a colony to grow, right? You need to lay eggs, you need to forage, you need to turn that food into workers through incubation and feeding. So every one of those steps is really important, and there's evidence that just about every one of those steps can be impacted. So yeah, among those are direct exposure to the larvae for sure.
So, do the bumblebees dance?
That's a really great question. They don't dance in the same way that honeybees dance. So they don't have this sort of sophisticated communication that I'm sure many folks will be aware of, where honeybees will come in, if they've found a good resource, they'll encode not only, hey, there's food outside, but what direction do you need to fly to get to that? And what's the direction against the sun, and what's the quality of that resource, right? So there's actually, in that waggle dance, there's a lot of different avenues of specific kinds of information. So they don't do that. They do communicate about food, though. And so they communicate some much more basic aspects of food. So they communicate, they can get other workers within the nest excited. So like foragers will come in and basically like run around the nest and just bonk other workers a lot. This is one of the reasons I was saying in passing, just when they're in physical contact, we actually think that's an important mode of communication. We think that that's true, so they can activate each other. And it turns out the scent of the food they're bringing in, those workers that get activated will learn. So they're passing passing information about there's food, and and it smells like this, exactly. They're not communicating it's, 270 degrees from the sun, and here's how far it is. But it turns out also, honeybees don't always use the waggle dance. So this is an interesting,
And that was a big controversy back then.
Yeah, and they definitely use it, right? But it turns out it's one of multiple modes, and they also have a mode of communication that is a little bit more like the honeybee, more like the bumblebee mode of communication, and they're actually always deciding whether or not to use their own sort of foraging experience versus like listen to their neighbors. So it's interesting. There's just lots of modes of information that they're weighing, and we think that maybe essentially what has happened is that the honeybee waggle dance is a sort of evolutionary articulation on top of this more simple version of communication.
So I have one more, which might be too complicated for a short answer, but I'm interested in the genetics of this. So one queen has many, many eggs impregnated by one sperm donor,
Usually.
and yet you get inter-individual variability across each individual honeybee, or bumblebee.
But it's probably true to some degree in both. No, that is a great question. So, yes, that's right. So we actually see relatively low, we think there's relatively low genotypic variation within these colonies, and maybe more so than honeybees. So, because honeybees generally multiply mate, so there's multiple sort of, we would call patrolines. Essentially, there's multiple father's genetics in a honeybee colony. And the other component of this is, not to delve too deep into a field I know very little about, which is genetics actually, but because of sex determination in bees, which is not like us, it's haplodiploid. So if your male bumblebees have one copy of all their genes, of all their chromosomes instead of two. So there's actually very high, basically all the sister workers in a colony have exactly the same father, so the genetic relatedness is even particularly high, even given the low sort of parental diversity.
Can you say that again?
Yeah, sorry,
All
that's
male, I mean, all male bumblebees are haploid?
Yes, so instead of having a sex chromosome, they're unfertilized eggs.
And then do these
they have one
haploids
copy.
produce sperm?
Oh yeah, they are fully functional, they don't do as much as the
Most males behave as if
workers.
they have only
But,
half a step
yeah,
anyway, but.
they're physiologically capable organisms from a totally haploid genome.
So you're getting spermatogenesis without meiosis in these haploid
Yes.
bees? Whoa.
Yeah.
That's like what plants do.
Yeah, they're very strange.
Yeah.
So we have very low genetic relatedness, but it turns out, the other thing I want to say is, it turns out you have a lot of variation in the absence of genotypic variation, even outside of these. So I did my postdoc in a very odd lab for this work, but actually a really great lab in some ways, and particularly for this question, which worked on fruit fly personality. So turns out there's variation in fruit flies, right? Just like I told you about, there's variation in bumblebees, right? And in fruit flies, it's really interesting because you can create isogenic lines that actually have absolutely, like as close to clonal as you can get, rear them with absolutely minimal environmental variation, and they'll still have individual variation. So you'll still get righty and lefty flies. Essentially, I would say there's not a behavior that we've measured well in almost any insect that doesn't have stable individual variation. So we should probably think about that noise across individuals or variations across individuals maybe as like the rule, independent of, and genotypic variation can probably contribute on top of that, but it just seems like that shows up absolutely everywhere. Sorry, that was a little bit of a rabbit hole, but it's such an interesting,
Oh, way to go.
such an interesting question. Yeah.
Are neonicotinoids, neonics, bad for people?
Yeah, that's a great, so one of the reasons we shifted over to them is they do have much, much, much lower affinity for the mammalian version of the receptor. And so in general, the acute toxicity to humans is much lower at certain doses, and I think there's some evidence that I haven't really kept track with, so I can't speak enormously well to that. at certain levels of exposure to like farm workers, there might be some impacts on humans, but the primary health impacts are probably via nutritional and food system impacts.
Is there a standard bumblebee model organism species?
Yeah, as close as we can get to is the one that we mainly use, Bombus impatiens, the common eastern bee, common eastern bumblebee. So they're great because there's a few species globally that have been commercialized for tomato, or mainly greenhouse pollination, but especially tomato pollination. So they're commercially available, they're grown by a few companies. There's one here in North America outside of California that is available, another one in Europe, but there's a few others sort of under development. Actually, I think there's a third one in commercial availability in Canada now. But
Well, when did they become considered domesticated?
like in the past few decades, like I think
So
mid-90s is.
are they considered domesticated on a par with honeybee then?
Not on a par with honeybee, just because of the scale, the length of domestication, so it's much more recent, and also the scales of it are much, like I think the impacts of, at least if you think about it in terms of, certainly these species of bumblebees that are domesticated are 100% grown in facilities. They do not interact with wild populations anymore outside of when they bring wild queens in for genetic diversity. Yeah, so they're certainly fully domesticated in that sense, but there's a lot less of them. They've been bred for a lot longer. So like the impacts of sort of artificial selection are probably weaker, but probably also non-trivial.
Thank you.
Yeah.
So my question is along those same lines. If you start with the assumption that the pesticides may change the genetics of the bumblebee, how do you, with your research over time, make sure you're not moving the baseline of your your bumblebee population?
That we're actually doing selecting. Yes. Yes. So our bees don't ever get back out to the wild, so we exclusively work here. Although, no, I shouldn't say exclusively. We largely work on the commercially available species. We also do some bringing in queens and rearing sort of wild populations. And yeah, but those bees we don't then release to the wild, less for evolutionary selection reasons and much more for lab disease, especially since we have the commercial bees in the lab. We don't want to transfer those back out to the wild because that's a really serious issue. So our bees never make it back out into the population. But also in terms of just the scope for evolutionary impact, certainly the really big scope for that is going to be the absolutely widespread exposure out in natural environments. So we operate at the same concentrations that they're probably exposed to out in the field. Like, you know, we're operating in the parts per billion. That's, you put some compound on a seed, the crop grows up, and that's the concentration in a flower months later. So compared to the level that they're continuously being exposed to in the wild, I don't think what we're doing in the lab has much impact there. But it's a super interesting question about, so we actually know a lot, and there's some great labs here in our department thinking about pest evolution to the massive pesticide exposure over the past, half a century or so. And we know a lot about that in pests, like Colorado potato beetle, for example, is one of the model systems for that. We actually know very little about how bees might be evolving in response. So it's actually something we're starting to get interested in. We're starting to look a little bit, and one of my, I think I put a picture of him up somewhere, but one of my grad students is interested in thinking about how different areas with different histories of pesticide exposure might have differential sensitivity. Does that make them now differentially sensitive? So something we're thinking about, yeah.
Is there technology that you can stick a tag on an individual bee? Is there a way to sample it, to get a DNA sample or protein sample from it without harming it?
Yeah, there is actually great techniques. So some folks are pioneering this with bumblebees in particular, just being able to take you mentioned the tarsus earlier. It's tarsal clipping. So you can clip off just the end of one leg and do genetics on that. So I think, yeah, that's great. And then, so one reason for that you guys may know is around here we have one of the last remnant really healthy populations of one of the federally endangered bumblebees, which there's not a lot of, the rusty patch bumblebee. So that has catastrophically declined over almost its entire range. Right here, like at the Arboretum, is one of the healthiest populations across all of North America. And so I say that because one of the reasons folks were really interested in developing those kinds of techniques is to not have to lethally sample bees. And so now you can do genetics on these endangered species without, without having to take them out.
And how do you non-lethally anesthetize a bee?
A bumblebee, yeah, so that's easier. Either cold or CO2 are our primary ones.
So if you had cold beer.
Work great.
So we're trying to transition here to.
That was a smooth transition.
One more question then,
Yeah.
maybe to finish things up here. What do you think of the work being done to engineer little robot bees to help with pollination?
I should say, so some of my earliest work actually was, like I said, was in biomechanics, and was actually partially funded by a big grant that made it onto some of the list of like worst spent U.S. federal money. So I think there's huge value in those projects, especially for trying to microsize robotics, and I think that project had just remarkable value in terms of the engineering that came out of it, a bunch of cool techniques. As a replacement for pollinators, you're not gonna, I mean, like I said, there's so much about these organisms that we cannot replicate. The power density, the phenomenal neural control, the whole life cycle, right? So when you think about what it would take to get the technology to the point where it can replicate what we're already getting, I mean, just the costs are not even, we would probably never be able to do it, and the costs of getting there are so far beyond what we would need to just like keep the service that we have from the existing population. So I'm skeptical about the ability to, in any sense, replace what pollinators are doing, but I think there's a lot of cool technology that will have a bunch of other great applications. Yeah. Yeah.
My question is regarding the sleep cycle of insects like humans, right? Sleep about a third of their life.
Yeah, so super interesting question. There's some variation within bees, so not all of them actually have to really sleep. And so partially it's because, keeping the temperature up and keeping the larvae fed has to happen around the clock, right? It's like nurses, right? At a hospital, you often think about, little, so one of the really interesting things is it turns out the circadian cycle depends a bit on what task you're performing. So the nurses have a much weakened circadian cycle, and foragers have a much stronger circadian cycle. So actually in honeybees, which are different than this system, so bumblebees honeybees move from being nurses to being foragers during the course of their life very predictably. Bumblebees don't do that. But with honeybees, they start off basically without a circadian rhythm, and then that circadian clock circuit kicks in when they start foraging, and you can sort of experimentally force them to revert, and the circadian clock goes away. So it's really closely tied to what they're doing behaviorally.
So that question makes me wonder, how do, in a nest underground, I'm assuming, how do insects that go from being out in the full bright sun into a pretty dark, if not completely pitch black place, how do they navigate underground?
Yeah, that's a really good question. I don't actually know if we have a good answer to that. I mean, almost certainly olfactory. Honestly, probably their navigation, at least for bumblebees, is like not that complicated. It's essentially like usually an abandoned mouse's nest with a single tube connecting it to the outside. So I don't think the spatial navigation is that complex in some sense. So my guess would be tactile and then also olfactory. Like we know olfactory communication is super important within the nest. And that's probably, the picture they showed, where do bees like to be? I'm sure that a lot of that is that's not vision based. That is almost certainly olfactory.
Is there any evidence for infrared vision in bees?
Yeah, I don't think they get, we need to remember the options, but I don't think they get very deep into infrared. There's some in other insects, there's some evidence that they might be getting some IR through not actually through their eyes, I think, but not literature I know enormously well, yeah.
There you go.
Thanks. Yeah, could you say something about what the role is of drones?
We use them. Sometimes. Yeah, I mean, I think maybe in some ways similar. Are you thinking about drones as sort of substitutes for?
Those drone bees.
Oh, sorry. I thought you meant, yeah. Well, here they're the males, right? So, I mean, their role are flying sperm packets for the most part. There is some, I mean, yeah, so that's like, they're not doing a lot. They're usually not sticking around in the nests. Yeah, they do, there's some evidence that they might do a little bit of like help out on behavior. Sometimes like do a little bit of nursing. They're maybe not doing like absolutely nothing, but they're not doing much. A lot of what they're doing is, you see them at the end of the season here and they're out, yeah, right, foraging for food, and, they'll mainly like sleep on flowers, and yeah, so certainly in terms of contributing to the social work of the hive, they're doing a lot less.
Boy, I know what I'm coming back as. Any other questions? I have one. Is it time to adjourn to the library? Absolutely. Thank you very much.
Yeah. Thank you, guys.